Okay, back at it once again. So this is the last part of chapter 7. This has to do with traffic in and out of the membrane. This is probably one of the most important. In addition to knowing the major components of the membrane, this is probably the next biggie is how things get in and out of cells. So you're going to hear the term plasma membrane or cell membrane refers to the same thing. Anytime we're talking about internal membranes within anything, any type of cell, especially eukaryotic cells, membranes are membranes, meaning that they are made of phospholipids, proteins, cholesterol, and integral and peripheral proteins. That's it. They're all basically universal as far as their components. Some of them are tighter, some of them are looser, depending on where they are in certain living things, and they allow more or less stuff to get through. Okay, so some of the examples that we'll talk about. Obviously, you need to move things in and out of your cells in order to keep the cells alive. So whether it's getting food and nutrients inside the cell or getting waste and debris outside of the cell. We'll talk about how oxygen and CO2 gets in. We'll also talk about some of these important ions such as sodium and potassium, which run the sodium potassium pumps, calcium, which helps in bone growth and cell rigidity. We'll talk about chlorine and how that works as well too. So passive transport, just like the term applies, passive means it's very laid back. It doesn't take any energy for it to happen. It's slow. Okay. Passive transport. Anytime you hear the word diffusion, it's always passive transport. You've heard osmosis. You've heard diffusion. It's always going to move down the concentration gradient, which is another term you're going to see a lot. Down the concentration gradient means it moves from where there's more to where there's less. No energy required. The energy actually comes from the fact that there is more stuff in one place than there are in others. Okay, this will increase with temperature. So as temperature increases, the membranes become more fluid. Passive transport can happen faster because the spaces within the membrane get wider, or the phospholipids are moving more. Okay. Not only that, the molecules themselves are moving more. So if you're talking about trying to get, I don't know, food coloring into one of those dialysis tubes, if you heat up the water that's there, the food coloring is getting faster. Okay. So once again, passive transport down the concentration gradient. Here's a simple example, simple diffusion. Left-hand picture, you have water with a membrane, and there are molecules. They happen to use the same thing I just was talking about. You might have dye molecules. Let's say it's a red color molecule, and it wants to move from where there's more to where there's less. All right. As it moves across the membrane, if they're small enough, they'll move from where there's more to less until it's equal. This is simple diffusion or passive transport. This is just through a membrane. Small things can do this. Large things will not do this. Okay. Some terms. We're going to probably talk about these terms at nauseum in the next day or two. So hypertonic, hypotonic, isotonic. Hyper means more. Hypo means less. Iso means same. Has to do with what's called solutes. So in a solution, there are two parts. There's a solvent and a solute. The solvent is the bigger part of the solution. The solute is the smaller part. And solutes tend to be, for lack of a better term, solids. So imagine we go back to this here. There is a solvent, which is the water, and the solute are the molecules. All right. If I compare these two, on this side it's hypertonic. On this side it's hypotonic. Hyper meaning more molecules. Hypo meaning less. Still hyper, still hypo. Eventually they're isotonic, meaning they both have the same number of molecules on either side. This becomes hugely important when we talk about how cells regulate things in and out of their membranes. So here's an example of osmosis. Once again, osmosis is diffusion of water. Notice that you have these tubes here. This is kind of a classic one that you see almost in every textbook. So there's this tube. It's a U shape. It's got a selectively permeable membrane. I'm making the assumption based on this that the molecules, the pinkish or purplish molecules here, are too big to fit through the membrane. So because they can't fit through the membrane, the number of molecules stays the same on both sides. Notice that there is more water on the left than there is on the right. This one is hypotonic, means it has more water, less solutes. Hypertonic, less water, more solutes. You can physically see the fact that there's more space in there. So the molecules can't move, but so if the molecules can move, they move from the right side to the left side, but they can't. If the water can move, which it can, it'll move from the left to the right. Why? Water's going from where there's more to where there's less. The pressure of there being more water, physically more space, drives that movement. And this would be the resulting um, sample. So, so animals want to live in what's called an isotonic solution. They want to maintain a normal internal environment, and the way they do this is by pumping water in and across their membranes until it's stable. Right. Sometimes we use the term equilibrium for this. Okay. So isotonic equilibrium terms are very similar and used in conduction. Put a little asterisk on the slide. This is one you have to know. So gives us um, three possible scenarios: hypotonic, isotonic, hypertonic. You can't use the word hypo, iso, or hyper without making a comparison. It's like saying, I don't know, I'll use the example that I am tall, which we know is not true. But I can't use the word tall without having a comparison. The comparison would be somebody shorter. You know, if you take somebody who's I don't know, a fourth grader who's 5 foot tall, I would definitely be tall, hopefully be taller than them. All right, so there, you have to make a comparison. So the comparison being made here is between the cell and the solution that it's in. When you take in these animal and plant cells, obviously the major difference: water vacuole, cell wall, both have cell membrane. If you take a isotonic solution, so a solution that is equal in concentration to an animal cell, water will enter and leave at the same rate. Animal cells like to be in isotonic solutions. On the other hand, plant cells, not so much. This is not the normal for plants. Okay? When they're in a solution that has the same amount of water inside and outside, they tend to be, they use the term flaccid. Think of a plant that's wilting a little bit. Okay? Hypertonic solution. If you drink a salty solution, right, water with salt in it, now there's more water with salt outside your cells than inside your cells. Your body or your cells are going to lose water to the surrounding environment. A good example of this is taking a fish that lives in fresh water and putting it in salt water. It's not used to all the hypertonic or salt water around it. Their cells literally get smaller because they're not able to maintain a balance of water inside their cells. So this is not good for most animal cells. They want isotonic. Conversely, hypotonic. If we took a saltwater fish and put it in fresh water, now there's more water outside, less solutes. Water's going to rush into the cells of the fish, and it's actually literally going to cause their cells to burst or lyse. Okay. Notice that in no picture here do the plant cells burst or lyse, and the reason they don't lyse is because they have a cell wall on the outside. So in the case of plant cells, normal is hypotonic. They want to be in a solution where there's more water, less solutes, and this is what happens. They get rigid or turgid. This is another term they use for it. If you think about the plants near the side of the road after the winter, tons of salt they use on the road to keep the drivers safe so that there's no ice on the roads. It's a hypertonic, so you created a hypertonic solution. More salts, less water. What happens to the plants in that area? They shrivel, all right? They get plasmolyzed. So their cell walls stay roughly where they're supposed to be, and they shrink in a little bit, but their cell membranes get super small. Notice the size of the water vacuole. It changes in these different situations. Osmoregulation. This is kind of a wicked cool concept. So I love to fish, love figuring out how fish are going to live and what they're going to do. There are some really cool fish in the ocean, or fishes in the ocean, that these fishes have the ability to regulate the water in their body. Okay, you see it with a lot of organisms that live on the edge, for lack of a better term. So if you're someone, if you ever watched the movie Finding Nemo, all drains lead to the sea. That is pretty correct. All right. If you think of bodies of water in and around East Bridgewater, many of those bodies of water you can trace back to rivers and streams. You can chase those rivers and streams back to marsh areas, like towards the coast. And if you keep following those marshes, you end up out into the ocean. So there are these areas between freshwater and saltwater that are called brackish waters. Okay, they tend to be estuaries. A lot of the organisms that live there they have the ability to regulate their internal water environment. They've adapted, they've evolved to live in those areas that aren't too salty and aren't too fresh water. But if it does get salty, they're okay, and if it does get too fresh, they're okay. So types of molecule movement through membranes. So one such example is facilitated diffusion. Facilitated diffusion is a type of passive transport. You guys modeled this, or hopefully one of your groups did. All right. So there are these molecules on one side. These are the solutes, or the things that need to be transported across the membrane. They're evidently too large to get through the membrane. So because they're too large, they're able to travel through these tunnels in the membranes. Remember earlier we saw a picture of a membrane protein with these like little loops or alpha helices in there, right? That's how these things actually get through, okay? Many of these channels are not gated. They're just open. So they're open to anything that's small enough that can fit through, okay? Has some good and bad qualities with that. They'll keep moving till they reach equilibrium, till they're equal on both sides. One example, a specific one, is called an aquaporin. These facilitate large amount of water to get inside. Some work like enzymes, okay? And by enzymes, I mean that they're sometimes called gated channels. So this, if you look at this particular channel, it's open to the outside. As soon as the right shaped thing fits inside, it's like little hungry hippo guy. It changes shape and drops it on the inside, and then it's going to instantly open back up to the outside, and it will keep doing this, open and close and open and close. All right, as providing that the right molecule fits inside. Once again, moving from where there are more to where there's less, but the protein works specifically as a gate and only lets certain things in. Okay, cool. Active transport. Active transport is the opposite of passive transport. In active transport, it's always with a protein. It's always against the concentration gradient. So it's going from where there's less to where there's more. Okay, always requires ATP, which we'll really get into over the next couple of weeks. So the number one example that you're ever going to see, hopefully you did this as part of your presentation, sodium potassium pumps. Sodium potassium pumps are the most important protein pump or a protein molecule that's found in all cells. They allow cells to react to change. What they basically do is they pump sodium out and potassium in to make a difference of charge, and that difference of charge is then allowed to diffuse back into the membrane. This is how muscles fire. This is how nerves react. We're going to spend a lot of time on this in class. Hopefully you have some good questions. Here's a great example of sodium potassium pump. So we'll start here, number one here. First thing that has to happen is there has to be sodium inside the cell. Sodium inside the cell binds to these three molecules right here. So this is a type of gated protein. Okay, how the pump works, it doesn't change shape until ATP donates a phosphate to it and then changes the shape of this molecule. As it changes the shape, it opens up to the outside and it literally forces the sodium out. Notice you get these little triangles on one side, and ironically enough, potassium is a little triangle looking shaped thing here on one edge. It's a square, but kind of triangular there. Fits right into that little spot right there. As soon as two potassiums fit inside, the phosphate falls off, potassium comes back in. This phosphate is then going to be eventually added back to ATP or ADP at some point during the cell's cycle. Okay. This goes on over and over and over again. Eventually what happens is you get all kinds of sodium outside and all kinds of potassium inside. What does it want to do? It wants to diffuse in and out. When it diffuses in and out on its own, sodium and potassium, so if all the sodium from the outside comes back in, or till it's equal, and the potassium leaves till it's equal inside and out, you end up getting muscle contraction and nerve reactions. Okay, here's a great comparison. So diffusion, simple diffusion. Movement of molecules from where there's more to where there's less, across a membrane, no energy required. Facilitated diffusion, more to less through a channel protein, no energy required. Active transport, less to more, uses ATP. All right, I always think of like your basement if you have water. Water's diffusing from outside to inside, from where there's more to where there's less. Okay, you don't want it there, so what do? You turn this pump on that you might have that requires electricity and sends it from where there's less to where there's more. All right, so kind of interesting scenario there. So there's this thing called electrochemical gradients. I'll get with more in class. membranes have a potential energy, all right, and it's rated in voltage. We can actually measure this. If you've ever had an EKG before or any type of um, encephalogram, sometimes they do one where they put little leads on your head. Sometimes they'll put leads on your wrist or your finger, and they can actually measure the membrane potential inside your cells for different medical procedures. Okay, we'll talk about this a little bit more in class. I think we can draw some great pictures to go with this. All right, where you see this most are these things called proton pumps. Proton pumps are found in primarily in chloroplasts. It's also going to be found in mitochondria. Um, the idea of the proton pump is it's going to pump protons from where there's less to where there's more. Okay, which creates which creates a differentiation of charge. So we have positive on one side, negative on the other. Those charges then want to diffuse back against back with the gradient. So let's say on the inside, on the cytoplasm side, it's all negative. On the outside, it's all positive. Those charges want to move from where there's positive in and negative out until they're equal. All right, which is a type of diffusion. All right, we call that chemiosmosis or chemidiffusion. Cotransport. Sometimes we can pump a molecule one direction against its concentration gradient, so then it can help carry something else inside. This is really common with sugars. A lot of times we want to take in large pieces of sugar. The only way we can do this is to co-transport it. So I think of the analogy of, hey, you use some gas to go to school. You go in there anyway, and you pick up somebody else up along the way, and they ride with you. So you still have to use energy to make it happen, but two people get there as opposed to one. So the same thing is happening here. So proton pumps, they charge the membrane by pumping protons or hydrogen ions across the membrane, and then they're able to then, when they diffuse back in, they diffuse through a protein, and they help sugars get in as well. Endo and exocytosis. Endo is in, exo is out, pretty self-explanatory. Endocytosis is the cell's going to surround things and pull it in. Exocytosis is going to release molecules from the inside of the cell out of the cell with vesicles. All right. This was another one of these pieces that you could have done. Here's an example of phagocytosis, which is a type of endocytosis. All right, so endocytosis or cell eating, which is phagocytosis, Phagoctosis, or phagocyte, which would be the stuff here. All right, so when the cell would make these tiny little pseudopods or little extensions, and the way they make these extensions is they use the cytoskeleton. It'll wrap around that food, it'll then pinch off, and now you have a new piece of membrane around there, which is then a food vacuole, all right, which can then, we know, go to the lysosome and kind of go from there. Here's a cool electromicrograph or electron microscope picture of that. Penocytosis, the same exact thing as phagocytosis, except they take in large quantities of liquid. No difference, just instead of being a solid, it's a liquid. Okay, works the same way. Cell moves around a body of liquid and then pulls that back inside. Receptor-mediated endocytosis. So mediation means to help. All right, so in receptor-mediated endocytosis, sometimes we bring things inside the cell, but we need to know what we're bringing in. And we have these things called ligands. All right, ligands are special receptors and binders on the outside of the cell that are actually going to help cells decide what they're going to bring inside. Here's an example of that. So here we have these receptors on the outside. All right. Notice the receptors are they don't care about the round things out here, the little green things. All they really care about are these ligands here. These ligands are the things that are going to be pulled into the cell. So what's going to happen is when those molecules are there, they bind to the receptors, and the receptor then says, okay, sends a signal inside the cell that this is the thing that we want to take inside. So it starts to fold in, make this little indentation in the cell. Okay, it also starts to coat the inside of the cell with special proteins so that we know what's in there. Okay, notice along with that, some of those green things get in there as well. So the receptors mediate what is going to come in. Receptor-mediated endocytosis. All right, good place to stop. A lot of questions. I'm hoping you watch these before class, and with any luck, we can crush this tomorrow. I'll talk to you later. Bye.